So the purpose of this unit is to really tie together the concepts of excitability with the structure of the neuron. And we can do that because different regions of the neuron have very different excitable properties. So very broadly, we can classify electrical signals as being either passive electrical signals. That means that they produce a transient change in membrane potential that's specific to one region of the cell and only occurs for a very short period of time. Graded and synaptic potentials are examples of passive electrical signals. On the other hand, active electrical signals create changes in membrane potential that can spread over a very long distance. An action potential is an example of an active electrical signal. Now, up to this point in the course, we've been describing the changes in membrane potential as being more positive or more negative. I want to complicate things a little bit by adding some new terminology in this unit. When we have a membrane at rest and it experiences a positive change in membrane potential, we call that a depolarization. And when that membrane returns to rest from depolarization, it's known as a repolarization. There's also a term to describe a membrane at rest, which experiences a negative change in membrane potential, and that's called a hyperpolarization. So in order to understand pretty much anything beyond this point in the lecture, you're going to need to be able to apply these terms: depolarization, repolarization, and hyperpolarization. So here's another cartoonized view of a neuron representing each of those regions that I just talked about. We're going to work through the rest of this unit by talking about what happens in each of these regions. And what you're going to see is that dendrites are typically the region of signal input to the neuron. The soma is the place where signals are integrated. The axon hillock is where the action potential is generated, and it spreads through the axon over a large distance into the axon terminal. The axon terminal is where the synapse is formed, and the signal gets relayed to the next cell, which again could either be a neuron, as in most cases we'll talk about, or some other cell type, as we'll talk about towards the end of the course. Now, here in this diagram, we can see close up what a synapse actually is. It's a point of communication between two different cells, and it's separated by a really narrow space. In this example, we're seeing the axon terminal of a cell that's sending a message, and the plasma membrane on a dendrite in the cell that's receiving the message. In other words, there's a presynaptic neuron sending a message to a postsynaptic neuron that's receiving the message. And you can see from this picture that the message is chemical. There's a neurotransmitter that gets released into that synapse. And really importantly, in order for the postsynaptic neuron to receive the chemical message, it has to have ligand-gated ion channels on its dendrites. That means that we have receptors for the neurotransmitter that's being released. If it didn't, there would be no way for that postsynaptic neuron to receive the message. So in response to the neurotransmitter binding, ion channels will open or close, and that creates a change in membrane potential. And we call that a synaptic potential. Now, synaptic potentials are really just an applied example of the GHK equation. Because remember that neurons are mostly permeable to potassium and a little bit permeable to sodium and a little bit permeable to chloride at rest. And that gives them a resting membrane potential that's typically somewhere around minus 70 millivolts. But when those ligand-gated channels open or close on the dendrites, it changes permeability. And that causes a shift in the steady state away from resting membrane potential, and it follows all the rules that we've outlined in our last unit. So to try to emphasize the properties and the location of synaptic potentials, there are really two terms that we can use to describe the changes in membrane potential that they can make. An EPSP, or an excitatory postsynaptic potential, describes any positive or depolarizing change from rest. From the GHK equation, we might guess that an EPSP could be produced, for example, by opening up a sodium channel. IPSP stands for inhibitory postsynaptic potential. that describes any negative or hyperpolarizing change from resting membrane potential. And again, from the GHK equation, we might predict that an IPSP could be produced by, for example, opening up a potassium channel. Now, probably the most critical thing to remember about synaptic potentials is that they're a type of graded potential. So graded refers to the fact that these changes in membrane potential can have a variable amplitude. They can be really weak or they can be really strong. Synaptic potentials are also decremental, which means that they decrease their intensity over time and over distance from their starting point. And that's because when a neurotransmitter binds to a channel on the dendrites, ions are moving into or out of the channel through diffusion, but only from that spot. So remember, movement through channels is passive, and it always follows the rules of diffusion. So the image that's on the left-hand side of this slide is trying to show you ions moving in through a channel and spreading out from that point. Now, as the ions diffuse further away from the membrane, the change in membrane potential that they're able to create becomes weaker. Because remember, your cells are like the ocean, and membrane potential is a very localized effect. The image on the right here is showing you the changes in membrane potential recorded at different locations on the dendrites. The arrow represents the site of neurotransmitter release, and so you can see that if you measure right at the spot where the channels are being opened or closed, there's a relatively strong EPSP. It has a relatively large amplitude. If we measure other locations on the dendrite that are further away from that site where a neurotransmitter is being released and those channels are being opened or closed, there's less of a change from rest. And that's really what it means to be decremental. Now, the problem with all of these cartoons is that in order to try to show you the concept of synaptic potential, they only represent a single channel opening or closing. But in fact, there's a lot of neurotransmitter being released onto the dendrites at any given time. And therefore, there are many channels that are opening or closing all at the exact same time. So sometimes we'll be making EPSPs through some channels and IPSPs through other channels. And so it's really the job of the soma to try to integrate or make sense of all of those changes in membrane potential that are occurring at the exact same time. In a way, this makes the soma kind of like a supercomputer. It's tasked with adding up all of these changes in membrane potential. And you can appreciate how complicated that calculation must be if you look at the density of all the dendrites in this photo here, which is again taken from a slice of the brain. So the process of adding up all of the graded or synaptic potentials is known as summation. And summation is essential for starting the action potential. In other words, a single EPSP is just never going to produce enough of a change in membrane potential in order to spread from the dendrites all the way to the axon. That's because of limitations of diffusion. It's always going to take many, many, many channel openings or closings.